Good morning, everyone. Let's get started. We will finish lecture 4 today, and I think we will have a little bit of time at the end to talk about the lab that is going to be happening next week. So stay tuned for that. Last lesson, we were talking about the various functions of the membrane proteins, right? We know now that on the plasma membrane, in addition to the phospholipids, the cholesterols, there are also proteins in them, right? The integral protein, the peripheral proteins, and they have various functions associated with it, right? Attachment to cytoskeleton, cell signaling, cell recognition, so on and so forth, right? And one of the functions is to play a role in transport. And that's what we are going to be focusing on today's lesson. How things are transported into and out of the cell. Sometimes they just happens naturally. Other times it will require the help of some kind of membrane protein. Now, before we get into that, we're going to take a few moments to learn about the cellular energy, like where cell obtains its energy and how it's going to be spending this energy. And that brings us to this molecule that we have been talking about, for a few times now. It is called the ATP. In the body, all the reactions that are going on in a cell, we typically refer to them as metabolism. Okay, so that's all the reactions that are taking place. And we can categorize these reactions into catabolism or anabolism. Okay. Catabolism is also known as catabolic reaction. Catabolic reaction. It's just one is noun and the other is adjective, right? I guess. And anabolism can also be called anabolic reaction. Okay. So catabolism, the word kata is same root word as catastrophe. In catastrophe, everything breaks down, right? So that's what happens in catabolic reactions. You are trying to break down molecules into smaller units. That's something that we talked about in lecture 2 already. We have macromolecules. We can use hydrolysis to break it down into monomers. So our body does that as well. On the other hand, we can build things from monomers, and that would be anabolism. Typically speaking, breaking down molecules will release energy, whereas building things would take energy. Right? Think about the game Jenga, right? Those blocks, you probably have seen it before, like they're wooden blocks, and then you have to build a tower of it, right? So building that tower takes time, takes energy. Okay, but knocking it down is easy, right? You pull the wrong one out, the whole thing comes down, right? So in catabolism, when you break things down, energy is released, just like the falling of the blocks. But to build it back up, it takes energy to do so. So in the body, um, when we eat food, when we consume food, the food molecules are basically the macromolecules that we have learned, right? There are carbohydrates in the food that we eat, such as starch. There are proteins in it, right? Let's say you eat a slice of pizza, the cheese it's protein, right? And there are lipids in it as well. So all these big molecules, we need to break it down. And that's where the catabolic reactions, catabolic pathways are going to be essential. Okay. In the process of breaking down the food into its building blocks, right, it will release useful form of energy. These energy will be captured by ATP, right? The usable form of energy in cell. Right? I told you there are many different forms of energy in cell. The currency for expanding energy is in the form of ATP. Right? So by breaking down your food, you capture as much energy as possible using ATP. Whatever you do not capture is going to be lost as heat. Okay, we are actually not very efficient as humans. A lot of the energy are lost as heat. You can think of efficiency like a light bulb, right? Remember those you might still have this at home, right? The really traditional kind of light bulb, the incandescent light bulb. Those are very inefficient, right? The function of a light bulb is to generate light, but if you touch them, it's very hot, right? So it's only about 10% efficient. 90% of the energy, the electricity, is lost as heat, right? Nowadays, we have LEDs, which are much more efficient. You touch an LED light bulb, it's not hot, right? So we are kind of like those old light bulbs, right? We waste a lot of our energy as heat. Now, it's not all bad because we are warm-blooded organisms, right? We need to maintain a body temperature of 37 degrees, right? So that's what the heat is for. It keeps us warm, right? After a meal, especially on a cold winter day, you feel warm. You feel, nice and warm because of this. So now we get all these building blocks, right? So I'll just give you an example. Like in the food, right? We might have proteins. Okay, and again, like carbohydrates, lipids, and all that stuff, right? So after you break it down, you might have glucose, you might have amino acid, so on and so forth, right? Now we can take these building blocks and create molecules that are needed in the cell. Maybe you need to create a new proteins in the rough endoplasmic reticulum. So where are you going to get the ingredients? Well, you got it from breaking down your food, right? And now you build whatever protein you need. And that is anabolic reaction, anabolic pathway. And building things, like I told you, requires energy, right? So you basically capture the energy in the form of ATP, and then you use that ATP to build what you need. In other words, this is the most important line, right? In other words, the energy released from catabolism drives the anabolic reactions. Okay, without the catabolic reaction, we won't be able to build anything. Catabolic reaction provides energy as well as raw material that are necessary for the anabolic pathway. Is that okay? So one of the major catabolic reactions that are happening in the body is cellular respiration. We mentioned cellular respiration a couple times already. It happens in the cell where you take glucose and combine it with oxygen to produce carbon dioxide, water, as well as energy, which would be captured by ATP, as we know now. Where does cellular respiration occur? Which organelle is responsible for carrying out cellular respiration? Does anybody remember? No, nobody remember the powerhouse of the cell?
Mitochondria.
That's right. That is the mitochondria, right? So you can see in this picture, it's generating a lot of ATPs, right? So that's what we will be learning in lecture 5. So what is ATP? ATP stands for adenosine triphosphate. Tri means 3, right? Tricycle has three wheels. Triangle has 3 sides. Triphosphate has 3 phosphates to it, right? And the adenosine here is the same adenine as the one you find in nucleic acid. Remember in DNA we have A, C, T, and G. The A is the adenine, right? So in an ATP molecule, we got the adenine, we have the ribose sugar, and 3 phosphate groups here. Okay? The bond that's holding the last 2 phosphate is very energetic. Okay, so like we say high energy bond. Right? And it's a little bit unstable. All right. So what happens is you can add a water molecule and break that bond off, and it just release all these energy that is available for use. Once you remove the last phosphate, the molecule becomes adenosine diphosphate. Right? Only 2 phosphate left, right? So you see here, 3 phosphate, you chop up the last one, you have a free phosphate on its own, and then you have the ADP, and then you release the energy. And you can use this energy for various things, right? We're going to get into it in a bit. But like I said, you can use it to build molecules. You can use it in anabolism. So essentially, you have a lot of ATPs in the body. And anytime you use them, it will get converted into ADP. So that's like the fully charged version, and this is the drained version. And in order for you to create more ATP, you're going to need to have more energy from the catabolic reaction. Remember, these are reactions that is involved in breaking down your food, right? So in other words, as long as you're eating, you will have energy to recharge your ADP, right? So you recharge your ADP, it becomes ATP, you use it for whatever, and then now you need to recharge it. So it just cycles back and forth. It's like your phone, right? You use it and then you recharge it, and you need to use it. You can use it again, and then you need to recharge it. So exactly the same situation here. But instead of plugging the phone into the socket to recharge the ADP, we need the energy from the catabolism, from the reactions that involves in breaking down our food. Okay. So what do we? Yeah, sorry. Go ahead.
So the catabolic is the one that breaks down food, right?
That's right, and that's going to be providing the energy to recharge the ADP. Okay.
Okay, thank you.
Yeah, so over here, right, it says the catabolic pathway releases useful form of energy, but we need to capture it. If we don't capture it, all of it is just going to be lost as heat, right? So we need the ADP to be there, and we're basically storing the energy until we need it. So what do we need it for, right? Well, we can use it to do different kind of work. Work basically means energy expenditure, right? So we need it for mechanical work. Mechanical work means to move something, right? So cells, they can move on their own, but as an organism, like we have to move around, right? To talk, to move the hand, to walk around, all that movement requires ATP, right? That is called mechanical work, all right? Without ATP, your muscle will stiffen and then you won't be able to move, right? And that's what happens when a person dies, right? After death, you are no longer breathing, of course, and without oxygen, you cannot do cellular respiration, and then you won't be able to make ATP anymore, and then the person will stiffen up, right? We call that rigor mortis. You might be familiar with that. Yeah, thank you. Someone just actually typed me the name. Right. The second function is transport work, right? Like I said, sometimes things would just move on its own across the cell membrane, but other times they don't really want to do that, okay? Maybe it's too big, maybe it's against is concentration gradient. We'll talk about what that means. So there are instances where the molecule just don't want to move. So you have to use ATP to kind of pump them over. And finally, we have chemical work. Right? So we already talked about how we need ATP to build molecules, right? If you take amino acids to make proteins, that requires energy in the form of ATP. And then there is the case of enzymes, right? I taught you enzymes a while back. Just want to see if you guys remember how does enzyme work? Enzyme works by lowering what? That's right, Roy. Thank you. Lowering the activation energy, right? Remember, I used the analogy of a tuition, right? Like tuition is too expensive, so the enzyme is like a scholarship. It lowers the tuition, so it's more accessible. People can pay for it, right? But here's the question: Now let's say the tuition is $5,000. Okay, scholarship is $2,000. So you only pay 3,000, right? But the scholarship is not free, right? Someone has donated that money, right? So that's the same idea here. Even though the reaction requires less energy now, something has to make up for that difference, right? Like where is it coming from? Well, now we know. In some cases, it comes from ATP. All right. So let's go back here for a second. Right here, right? So energy for anabolic reaction, but not just that, right? We can also use it for like mechanical work, for transport. So just keep in mind that there are many uses of ATP within the body. All right, let's do a quick poll and see where we're at. All right, last call. Anyone else? Great, thank you. So catabolic reactions or catabolism is breaking down your food, right? So let's see, consumes more energy than they produce. That's not true, right? When we break down the food, we try to extract energy from the food, right? If it takes more energy to break down the food than they produce, then that's like eating would be disastrous, right? Like it would be you would be losing energy from eating, right? So that's not true. Breaks down polymers into monomers. Yeah, that's essentially what breaking down means. Produces excess heat to warm the body. It was in the diagram. And of course, it happens in other organisms as well, not just human cells. All right, we're gonna take a look at transport now. Right? There are many different kinds of transport that occurs across the cell membrane, and we will start with a very simple type of transport first. It is called diffusion. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. For those of you who have a hard time understanding concentration, think of two beakers, okay, 2 cups, same volume, okay, same volume, same amount of water, and then we're gonna add salt in it. I add a lot of salt into one, and then I only add a little bit of salt here. So this one is high concentration. High concentration. We use square bracket to represent concentration, okay? So very salty, basically. And over here, because we did not add as much salt, this is lower concentration, okay? Now, the key thing here to know is that they have to be the same volume for you to have meaningful comparison, right? Obviously, if I added more water in here, I would be diluting it, and the concentration would go down, right? Similarly, if I evaporate some of the water by boiling it, then this would get more concentrated, right? So if I have a tube that connects them, and I allow the water to flow between the two containers, diffusion means the salt is going to move over, right? Going from high concentration to low concentration. Okay. The picture here shows you visually what's happening. You can add a few drops of dye into a flask of water, and you don't have to even do anything. You just let it sit there. It will slowly spread out until the entire flask is going to be blue. Right? So this is diffusion. The dye is very concentrated. There are no dye in other area, so it's spread from high concentration to low concentration. This is a spontaneous event. Excuse me, this is a spontaneous event, meaning it will happen on its own. You don't have to do anything to promote it. I mean, you can make it faster. You can swirl the bottle and whatnot, but you know, on its own, it will happen. Okay, and it's going down their concentration gradient. A gradient is basically a difference between 2 points, between 2 areas, you can say. All right, so as long as the concentration is different between 2 area, it will naturally go from high to low. Okay, one last example. You know, if this was a classroom, okay, and then I open a bottle of perfume at the front, right, or like for bees, right, you spray that stuff, okay, at the front of classroom, people will be able to smell it at the back later on, right? Okay. Very concentrated, and then just spread naturally throughout the room. So that's diffusion. Pretty straightforward, right? I hope. We're going to look at a few factors that affect the rate of diffusion. Right? So we know it happens on its own, but what would make the process faster or slower? First factor, the size of the molecule. Things that are smaller tends to move faster, right? So you would expect them to spread quicker. This is oxygen here, right? And this here is glucose. Right? So you would expect things like oxygen to diffuse faster than something that's bigger, like glucose. Second is temperature. Higher temperature causes faster diffusion. Think about like a hot summer day, okay? Like you walk maybe on the street and then it's garbage day, okay? You can smell the garbage, right? But on a cold winter day, you don't smell too much of it, right? Because higher temperature allows things to diffuse faster, right? To go all over the place, right? Okay. Next one is the steepness of the concentration gradient. Okay. Remember, gradient just means difference between the 2 areas. If the difference is greater, then the diffusion will happen faster. So let's say here, right, we have a lot of solute, a lot of salt. Let's say this is 80% concentrated, and over here it's like 0%. There's nothing, right? So the difference between these 2 areas is 80. Over here, let's say this is 60%, and then this is like 30%. The difference is only 30%. So you would have faster diffusion here, because the gradient, the difference is greater, right? A way to help you visualize this is like this. Think of a ram. Okay, you have this ram, okay, and then you have this ram, right? Which ball is gonna roll down faster? Ball A or ball B? Hopefully, based on common sense, you would know ball A is gonna roll down faster, right? Because this is a steeper hill, right? The difference in height is greater compared to the difference in height here, right? And that's essentially what's happening. The difference is so great, the diffusion will be occurring faster compared to when it's like a more gradual. It will still happen, like these things will still go over, but it's not going to be going over very quickly. Are we okay? Any questions so far? All right, thank you. Let's move on to the last thing. Now this one is a little bit hard to visualize, okay? Like even when I was explaining it in person yesterday to my class, they still don't really get it. So you know, if you don't understand, just let me know. So let me just try to move these things over. Okay, so here we have a bunch of H plus. All right. Light charges repel each other. Right? Positive don't like to be next to other positives. Negatives don't like to be next to other negatives. So if you have a lot of positives here that are in a concentrated area, this is going to be pushing each other away, right? And this is going to happen faster compared to something that has no charge. Let me maybe draw this out for you. You don't have to draw this, okay? So like if I just have a bunch of molecules like this, okay, versus this situation. Okay, so let's say we have two containers here, okay? Again, you don't have to draw this, you can just watch. Okay, and I make all these guys positive. For some reason, they're not showing up. Come on. So weird. Anyway, you understand what I'm trying to say, right? They keep on disappearing. So my question to you is, do you expect these to go over faster, or do you expect these to go over faster? Right? Now, hopefully, based on what I try to explain to you, the diffusion is going to be faster on the right side, right? Right? For these charged ones, because they have the charge. The positive don't like to be next to each other. They will be pushing each other, like, get out of my way. Okay, go, go, go. Right? So that repulsion force is what will be forcing them to move quicker along the way. Right? Over here, they'll go, but then they lack that repulsion, right? They don't have that extra push to cause them to go faster. Does that make sense? I don't know why this thing is not showing up, but hopefully you get the idea. All right, let me erase this stuff.
So it's the same with negatives one, negative, like negatively charged things as the positive.
Exactly, that's right, exactly. Now, if you have a mix of positive and negative, then that's a different story, right? If initially the mix contains both positive and negative, that would actually slow down the diffusion, right? Because then now they're attracting, right? Okay. But the key thing to remember is that, you know, as soon as you introduce some charge to the mix, it will affect the rate of diffusion. Okay, I'm gonna go through a concept with you here. Now, listen up, huh? A question like this will show up. I don't know, sometime either on your quiz or on your test. I don't remember where, but it will show up. Right? So make sure you understand what is going on. Okay, so listen up. So this is osmosis that we are going to be talking about. We're going to be writing a note as well. Okay, so get ready for this. Osmosis is the diffusion of water. Okay, diffusion of water. So let's start very simple. I'm gonna have a container here. Okay, and then I'm gonna separate it with a barrier. Okay, so this barrier only allows water to pass through. Okay, you can create different filters that have different pore size, so that only small things can pass through. Okay. Now I'm gonna fill this up with some water, okay? And then I'm gonna add some solute to it. Oh boy, that's too big. Let's all add these circles to it, and then just a few here. Now, can we all agree that the left side has higher concentration? Right? Because it has more dots, right, in the same amount of volume. And over here, it has low concentration. All right. I hope we can at least agree on that. Okay, so I'll let you draw first, and then we'll explain. All right. You don't have to erase anything, just listen. If I remove the barrier, which way will the red circle go? And just think about it. This is not a trick question. If I remove the barrier, which way will the red circle go? Is it going to go to the right or go to the left? Well, hopefully you understand it's going to go to the right, right? It's going to go from high to low. All right. Everybody should have no trouble answering that. Things naturally goes from high concentration to low concentration, right? Now I'm gonna put the barrier back. Okay, remember this barrier only allows water to pass through. So even though the red circles wants to go to the right, but they can't. The gate is down. They are too big to fit through the pores, right? They won't be able to go through. So in this case, only water will be able to move and not the red dots. Okay, only allows water to pass through, right? Red circles cannot cross. So here's the difficult question. Which way is water going to move? Okay, let's do a poll and see what you think. Which way is water going to move? If you think it's going to move to the right, please choose A. If you think it's going to move to the left, please choose B. If you think it's not going to move, please choose C. Which way is water going to move? Okay, well, we have a three-way split here. Okay, we have 5 people thinking is going to the right, the other 5 think is left, and then we have some who thinks no net movement. All right, so listen up. I'm going to tell you the answer, and you better understand, because like I said, there will be a question on the test or quiz related to this concept. Okay, let me ask you another question first. If you take a spoon of salt, and then you put it in your mouth and you hold it there for like, I don't know, 30 seconds, what's going to happen? If you don't know what's going to happen, maybe you try that during the break, huh? But try to visualize this. You put a spoon of salt into your mouth and you hold it. What's going to happen?
It'll start dissolving in your saliva.
Yeah.
Yeah, and but then what's going to happen to your mouth? Like, remember, we're not talking about like a few grains of salt, right? Like a whole teaspoon.
Teaspoon.
Your mouth will be numb.
Sure, your mouth will be numb. Your mouth gets dry.
Salty
Really?
dry.
Actually, your mouth would get very watery. Okay. You
Oh,
guys
that
should try.
makes sense. Yeah.
Right, like you would drool all over the place, right? Okay, like
Mhm.
it's not gonna be a good feeling, but that's what it is, right? Because the water is being drawn out from your mouth by the salt. Can you guys visualize this? Maybe, yeah. You can try this. Maybe don't do a
Yes, we
spoonful,
can.
just like do like a a pinch or something, right? And that's what's happening in this case. The water is being attracted towards the saltier side. Okay, so water is drawn. Oh, sorry, drawn to the higher concentration. Okay, to try to balance things out, right? That is why people do salting. I mean, before refrigeration was invented, to preserve food, you need to basically use salt to preserve them. Why does salting the food preserve them? Anybody? Yeah, it removes the excess water, exactly, right? And without water, bacteria is not going to grow, right? Okay, so when you put them in salt, it sucks out all the moisture in it. Okay, so that's what osmosis is. Osmosis is going to move towards the saltier side. Okay, so on the test quiz, again, I don't know when is it going to show up, but I
When
know
is it?
what's that?
When will it be?
After Thanksgiving, yeah.
Uh-huh.
So there is going to be a question like this. Okay, it's going to be on everybody's test because I can set it up like that. There's going to be a barrier here, and then there is going to be the same water level here. Okay, same water level. Okay, I mean it's in the PowerPoint slide, but try not to look at it. Try to think about what's going on here. Okay. And then we have the red circles, just like before. Okay. Let's put some here. Okay, so what's going to happen afterwards? What's going to happen afterwards? Okay. Well, based on what we have been discussing, right? The barrier will not allow the red circles to move, and the only thing that will be able to move is the water. So in this case, it's saltier over here, so the water level will rise on the right, whereas the water level on the left is going to drop, right? Okay. So lower water level, higher water level. And what does that achieve? Remember, the red dots does not change. Okay, so it's the exact same as before. Okay, but now, because the water shifts over, the concentration increase on the left. And over here, the concentration decrease on the right. And as long as the concentration is different, the water will be drawn to the other side. Okay. This is
But
called,
how is the water getting across the other side with the barrier?
the barrier is only going to be blocking the red circles, but the water molecule is small enough to pass through.
Get through?
Yeah. And so the water level will actually rise on the right. Okay.
Sir, when is the beetroot test?
I'm sorry.
Lab.
Yeah, you ask me that every class. Yeah, I'm going to talk about the lab towards the end. Okay.
Okay, great.
Yeah. So people who are diabetic, sorry, not diabetic, people who have kidney problems, okay, not necessarily diabetes, but people who have kidney problems, if they are waiting for a transplant, the only way to keep it functioning while you wait for the transplant is to do dialysis, right? Dialysis. Dialysis involves taking the blood out of your body and then filtering it, right? So the concept of osmosis is used, right? So it's actually there are these tubings with different pore size that helps you remove some of the impurities from the blood while you're doing the dialysis.
I have a quick question about the diagram.
Sure, yeah.
So the second one, why I don't understand why the concentration on the right is going up.
The concentration is going down on the right, but the water level is going up. Let me write that
Oh,
down for you. Yeah, water level goes up, but concentration goes down because more water is there, right? You're diluting it, right? By adding more water, right?
Oh, okay. I thought like yeah, that's
Sorry.
right.
Yeah. And over here, water level goes down. Okay, and this will continue to happen until the concentration are the same. Okay, sorry, I didn't know that was a confusion. Anyone else is confused, just ask. I don't know if you don't, I don't know what you don't know. If you don't tell me, if I'm not making sense, just ask. Okay, let's take a look at the one in the PowerPoint, huh?
Excuse me.
Yeah.
In the one position, concentration going to the right.
The water is going to the right.
Water. Yeah.
That's right. So in this case, right, in this particular picture, it's low concentration on the left and high concentration on the right, right?
After that,
After that, the concentration would be the same.
the same. Yeah.
That's right. Mhm.
But the same level of water, both
It's
sides.
at the beginning it's the same level, but afterwards you see the water moved up on the right side because they're
Mhm.
going to the left, right?
Yeah.
So as the water goes to the right, it will dilute the right side and then concentrate the left side.
Okay. Thank you.
All right. All right. I hope that makes sense. And again, just ask if you need clarification. We're gonna start a new note now. I don't know how much space you have, but if this is your paper, you should separate it into 3 columns. We're gonna write something here, here, and here. All right. So this is called tonicity. Okay. So basically describes water movement. When a cell is placed in different environment. All right. I'm gonna show you 3 different environment, and in each case, we will look at how the concentration are different outside and inside of the cell. We will look at the direction of water movement, and we will look at the end result of the cell. Okay? Start with the first scenario. This is called isotonic. Isotonic. So here is a beaker. Okay. We'll put a cell in there. Okay. And in isotonic, the concentration is the same inside and outside. Okay. So square bracket means concentration of the outside equals to concentration of the cytoplasm, the inside of the cell. All right, I'll give you a moment to catch up. So in this case, what's going to happen? to the water? Well, if there are no difference in concentration, then rate in equals to rate out. Right? The same amount of water coming in as the amount of water that's leaving. Okay? So we'll use red arrow to show water leaving, and then I'll use green arrows to show water coming in. Right? So you have the same amount of water coming in as it is leaving, which cancel each other, right? So there will be no net water movement. No change in cell volume. Okay. Now, if you have a hard time visualizing this, take a look at this analogy. I have a what do you call this? Like a basin, and then you have a water tap. I'm gonna turn on the tap. Water's gonna come in. It's gonna fill up the basin. Okay. And then I'm gonna poke a hole here. So water is gonna be leaking. All right. So there's water leaving the basin, and then there's water coming in from the tap. If they are the same rate, what's gonna happen to my water level?
It's gonna stay the same.
Exactly, no
Yeah,
change
same
in
level.
volume, right? Okay, and that's what the cell is doing here. It's not that there is no water movement, it's just that they cancel each other out, so there's no net movement. Okay?
This reminds me a lot of what I learned in engineering. It's inflow and outflow. The more
Exactly.
inflow you have, the less outflow you have. The more outflow you have, the more inflow you have. Therefore, water level will change based on how much inflow and outflow you have.
Exactly, right? So if you're talking about like, a mixing tank or something, right? Industrial mixing tank, you want the things that going in, going out is the same, so you can have uniform concentration, right? So that's essentially what it is. Very good. So like, is the cell happy or not happy? Well, if it's an animal cell, happy, okay? We love it. This is the best environment that an animal cell can ask for. If this is a plant cell, they're okay. Right? Not the best, but they're not dying. This is like if you forgot to water your plants for a couple of days, you see them, kind of become a little bit withered, not dead yet. You can revive them, right? But it's not the best. That's the easiest case. Isotonic. All right. Hope you're ready for the next one. Next one is hypotonic. I like you to put a big O here. Hypotonic. Hypotonic. Let's draw the beaker. So in this case, we have a cell here. And it's a lot more concentrated on the inside than outside. Okay. I'm just gonna write, I guess like on your page, you have it like right next to each other, right? Which, like I told you, to have 3 columns, right? It's much easier for you to compare them. So the outside in this case, oh, sorry. Trust me, I know how to spell. Outside is less than the inside, the cytoplasm. When I say outside, I mean like outside of the cell, right? is less than the cytoplasm. So in this case, which way is water going to move? Think about the U-shaped tube that we were talking about, right? Which way is water going to move? Into the cell or out of the cell?
Into the cell.
Excellent, right? So we have some leaving, but we have more coming in. We have more coming in, right? Okay. So rate in, rate, let's do the same order here. Yeah, rate in, it's greater than rate out. So net water movement is into the cell. And cell volume increases, right? Let's go to our lovely basin. Please think about this. To represent our situation here, should I turn on the tap higher or should I make a bigger hole so that it represent the situation I'm trying to?
Same.
What's that? Sorry.
The
Turn
same.
on tap.
Same. Turn on the top. Same
Turn
water.
on the tap. That's right. We're gonna turn on the tap all the way. Okay. So like like that, and then this is gonna be leaking, right? So we have more water coming in than leaving. What's going to happen to our water level?
Our level going up.
That's
It's
right.
gonna rise.
It's going to rise. And eventually what's going to happen, people?
It's gonna overflow.
It's going to overflow. Exactly. It's going to spill. So that's what happens to our cell. Eventually there will be way too much water in the cell. And if you're an animal cell, right, it will swell up and lyse. Okay? Lyse means burst. Okay? The animal cell will
Wouldn't that
basically
happen for the
explode.
same human cell?
Yeah, we are animals, right? So yeah, human cells, animal cells. That's right. So do you think we like it? Absolutely not. The cell's gonna die. Okay, this is the cell exploding, okay? Plant cell. Plant cell loves it. Okay. Will the plant cell burst? I mean, they will swell up all the same, but will they burst? Will they explode? Why not, Roy? They have a cell wall. That's right. Okay. So cell wall prevents bursting. Okay, they love it. You water the plant, right? They suck up all these water, store it in the central vacuole. We learned that last class. Pushes against the cell wall. All that extra pressure allows the plant to stand up. Okay, like again, if you don't water your plant, the leaves kind of get crumpled. As soon as you water them, they open up. Right? So this is hypotonic. The O, the fat O here is supposed to help you remember that they get really, really big. Okay? And it's possible to burst. Let me tell you a story. Do you guys know Wii? It's like a gaming system, right?
The Nintendo Wii.
Yeah, the Nintendo Wii, right? It's like an old thing now, okay? But you guys probably have heard about it. So when it first came out, I don't know, like what, 15, 20 years ago, probably 20 years ago, it was a hot thing. Yeah, everybody wanted to get their hands on it. And so this radio station in the States held a contest Okay, the contest was called, you don't have to write this down, I'm just telling you. It's called Hold Your Wee, as in your pee, right? Your urine. Hold your wee for a wee. Right, so that was the name of the yeah, some of you know about it. Okay, great. And you know, people participated, and this is how it works. The contestants have to drink as much water as possible and not go to the bathroom. And if you're able to hold it, right, last person standing, then you win the console, right? Okay. So a lady won. I don't know how much she drank, but she drank as much as she could. Was able to hold on to it, not going to the bathroom. She won the contest. She got the console, and she was found dead hours later in her apartment. This is called water toxicity. Okay, based on what I just teach you, why does drinking an excess amount of water in a short period of time, why is that lethal? Why could that potentially kill somebody? Too much to process? As in like too much information for you to process, or like too much water for the body to process? Oh, okay, so right. Okay, anyone else? Like based on what I just taught you here, why does drinking too much water in a short period of time, why is that potentially lethal? And you should not do it, no matter what they give you for free afterwards. Don't do it.
Well, your cell will like kind of explode, and then all of them will explode.
Basically, right? If you drink a lot of water, you are diluting your blood. Okay, you're diluting your blood. And you have red cells in your blood, right? Red cells are important for transporting oxygen. If they all started to burst because the blood is very hypotonic, you have nothing left to transport oxygen for you, right? That's one of the reasons why it kills you. Okay, it can also kill you because it's diluting all your electrolytes. You know, the potassium, the sodium, right? The chloride. You need those things for normal functions of the body, right? For the nerve to communicate with each other. And when you dilute it so much, then they stop communicating. The brain cannot tell the heart to beat, for example. Then you die. Okay? So that's water toxicity. You should not drink so much water in a very short period of time. Okay. That takes us to the third case. I hope you're still with me, honestly. I'm trying my best here. Hypertonic. Okay, hypertonic. We have a solution. And I mean, you can probably guess what happens now, right? It's the opposite. We have a high concentration on the outside and then low concentration on the inside. You can practically finish the note yourself, right? In this case, the concentration outside is greater than concentration in the cytoplasm. Okay, and again, I hope you're doing this side by side, and you can see it's just the opposite of hypotonic, right? So in this case, water will be drawn out. Okay, you have more water leaving than coming in. Right? So rate out is going to be, sorry, I gotta do it in the same order so that it makes more sense. Rate in first. Rate in is going to be less than rate out. Okay, so net water movement is out of the cell. Cell volume decreases. Okay. So here is our basin analogy again. By now, you should be an expert in drawing these water taps. So this time we have a super big leak. Okay, it's just like really draining. And then the tap is just normal. So you have the water coming in. Water coming in here. But then you have a lot of water leaving. Okay, so over time, I'm sure you can understand that the water level will be dropping, right? Okay. So what happens is the cell volume decreases, right? It shrinks. Okay, we have a fancy word for this. Plasmolysis. That's the biology term for shrinking. So if you're an animal cell, you are not happy, but like you're not devastated, like the hypotonic case. Like you'll live for a little bit, okay? Plant cell, this is bad. Okay, this is like if you forgot to water your plant for like a couple weeks. Right? It's really dried up now. It's on the verge of dying. So can someone give me an example of what you do to the body to result in this state? What's like a real life example? Is this okay? Okay, is this high enough? All right. Dehydration. Thank you. That's right. Right. If you don't drink enough water, right? If you you know, in a hot summer day, right, sweating excessively, then your body becomes hypertonic. Your blood is too concentrated. Okay. All right, ready for another story? I'm sure you are. So, I'm sure some of you drink coffee. I used to drink a lot of coffee, but then I quit. Anyway, but people like to drink caffeinated beverages. Okay, so other than coffee, people can also drink things like Red Bull, right? There is the one with the 3 green marks on. I forgot what it's called. Anyway, there are a lot of caffeinated drinks out there, right? So when Red Bull, oh yeah, Monster, that's what it's called, with the claws, right? When Red Bull was first on the market, people like to mix it with alcohols at clubs, nightclubs, okay? Because then, you know, you can party all night, right? With the extra caffeine, you stay awake, right? Okay. And this is a true story, right? So people add it to the alcohol and they drink it. But the caffeine actually makes your blood more concentrated. It adds more pink dots to your blood, so to speak, right? So it dehydrates you. I mean, you know this, right? Even if you don't drink Red Bull, like you drink your coffee, right? Like within an hour, you gotta go pee, right? Okay, and you can almost smell the caffeine in your urine, right? So it basically dehydrates you. Okay, so these people, usually younger people at the nightclubs, right? They add the Red Bull, they drink it, and then they feel thirsty. Okay, because of dehydration. And what they should have done is to drink water, right? Okay, but then that's not what they did. They're like, oh, I'm so thirsty, right? Let's drink more liquid. Let's drink more, you know, alcohol, more Red Bull. So they add it and then they drink it, and then they dehydrate even more. So by the time you do this couple times, like 3-4 times, your body is severely dehydrated. Okay, even though you're drinking fluid, but you're not drinking water to dilute the blood, right? And unfortunately, people also die from situations like that. Okay, so that's creating a very hypertonic environment for your cells. You are super dehydrated. And also for another reason, actually caffeine if you have too much in your system, sometimes it could cause arrhythmia. Okay, arrhythmia is when your heart rate is irregular. Okay, and it could cause you to have sudden death. You literally just drop dead. Okay, there are stories of these of like, you know, high school students who like drink too much caffeine. Right, Red Bull in the morning followed by an ice cap, and then, you know, more Red Bulls in the afternoon or something, and then their heart just couldn't take it. Right? So you gotta be careful what you put in your body. So that's isotonic, hypotonic, and hypertonic. I hope it makes sense. Why don't we take a quick break, 5 minutes? We'll come back at 9:41, and we will do more. Okay, see you in a bit, guys. Okay, let's continue.
Excuse me, Professor. I didn't get your record for last class.
You didn't get my recording for the last class?
Oh, I have
Yeah.
it right here. I can send a link.
Oh, thank you. Yeah, but it's all up here, right? It's the same place every time.
That's the lecture archive.
Yeah,
Yeah,
the
but
lecture.
I didn't see the link either.
You
Oh,
didn't
it's there now.
right here, right? It's Tuesday, September 26th. It's right
I
here,
just
right?
watched it last night, actually.
Okay.
Yeah.
I had just 21 September.
Say that one more
Oh.
time.
I saw just 21 September.
Oh, do you only see the 21st one? Okay, but it's there, right? You can see on my screen, right? It's right there.
Okay.
Yeah. Next time it's okay. You can email me if you
Okay.
can't find things. All right.
Okay. Yeah. Thank
Okay.
you.
Thank you for posting the link, Justine. It's in the chat now.
Yeah,
For those
I
of you who
did.
need it. Okay, great. Now I want to take a moment to talk about the lab, which is going to be due next week. Okay, so here is my plan. Like I'll tell you about the lab now, and I mean, you can start working on it. It's not going to be due until next Thursday, okay, which is the 5th. Okay, we are not going to have class on the 5th. You can just spend that day finishing up your lab. We will have class on the 3rd though. Okay, but 3rd, it's just going to be a shortened class. All right, we will finish whatever we don't finish today with lecture 4 on Tuesday, and then I will help you with your lab a little bit. Okay, but you got to work on it first, right? Between now and Tuesday, and then, you know, if you have any question, you can bring it to Tuesday's class, and then we can talk about it. Do you guys understand? Okay, so
And
no.
the test would be the next test would be.
Okay, so like again, you can look at the calendar, right? Do you know where to find a calendar, Georgia?
I think so.
Yep, so download the calendar and then you will know when things are going to be happening, right? Okay, so October 10th is when you're going to have the quiz. Okay, so again, just want to tell you, you come to class normally like Tuesday, but then no class on Thursday. You will just be working on your lab, and then you will be submitting the lab on that day. All right?
But for the quiz, we have to come to class.
Yes, for the quiz, you always have to come to class for that. Like you come to class normally, and then you would do the quiz like, you know, during the day, right? Okay, like before class, after class, at night, something like that. All right, so here is what you need to do for the lab. Let me switch to student view and see what you will see. If you go to content, and again, I'll post an announcement after class with links for you to click on. If you go to the virtual labs, there are 2 labs, right? There's the Beetroot lab, and then there is the microscopy lab. Right? So there is a demo that you have to watch. So this is someone else who did the experiment. I'm trying to create my own video, but that takes forever. Like I got to find someone to film me, and then we got to edit it together. I got the filming part done, but I just got to edit it. So like for now, you just have to watch someone else do the lab. Okay. It's a very short video, right? It is 8 minutes, right? you have to watch it in order to do the lab, okay? People in the past try to do the lab without watching, and they just try to search answer on the internet. I will know you search answer on the internet because your answer, like I told you, right? I've been teaching for like over 15 years, okay? I've seen all the possible answers out there on the internet. And if you just copy it from the internet, it would be likely wrong because the questions are tailored to this particular video, okay? When you find answers on the internet, they don't know you are watching this video, right? So the answers will be off. It's very simple. You watch the videos, the answers are all in there, okay? Most of them anyways, okay? I would say if you watch the video and then you answer the questions, you will be able to get, you know, minimum 70 to 75% of the answers on the lab, okay? So here, I'll even copy the video link and post it in the chat box. All right, there you go. Okay, but you can find it, right? You watch that first. It's a permeability lab, okay? We are looking at the purpose of the lab is to examine the effects of temperature on the membrane permeability. In other words, if you make it harder or if you make it cooler, is that going to affect how leaky the cell membrane is going to be? Okay, that's basically what the lab is about. Okay, so you watch that. And then you will have to download a copy of the Beetroot Lab. Okay, Beetroot Lab right here. So you click on that. Now, obviously, you cannot type in the question here, right? You cannot type it in. So what you have to do is you have to download it first. Okay, you have to download it, and then you can open it on your desktop. Okay. I'm trying to open that right now. All right, look like this. You're gonna enable editing. Okay. And then it says, what is the purpose of this experiment? I just told you what the purpose is, right? So you will type it in. The purpose, I don't know why is it not letting, it should let you type, okay? Let me try it one more time here. There we go. Okay, so you just go. The purpose of this experiment is to examine the effects of temperature on membrane permeability of beetroot cell. Okay, you're gonna be using beetroot. That's it, that's 2 marks. Okay, I give you permission to copy that. I give you permission to plagiarize me. Okay, 2 marks. You're welcome. Right? That's all you have to do. Okay? Don't make things more complicated than it needs to be. Okay, don't write me a novel. Just answer the question. Okay, don't use like big words unnecessarily. Do you understand what you need to do? Okay.
Yes.
Write a hypothesis. I told you, an hypothesis is not a question. Many of you wrote that on the quiz, right? Like it was the question about like driving with the window down or up, right? Okay. Don't ask a question for the hypothesis. It has to be a statement. Driving with the windows down will increase fuel efficiency. That's it. Okay. Don't explain why you believe that. Don't start telling me about the air conditioning and you know all this unnecessary information. No explanation required. It's just what you think is true. That's the hypothesis. You write it out. That's it. Okay. So I will tell you more about the lab on Tuesday, but you have to watch the video first. You have to try to answer some of these questions. Now, some of them are a little bit like hard and weird, and that's okay. I will talk about it on Tuesday. Okay, I don't want to talk about it now because you guys haven't watched the video and you don't really understand what's going on, right? Okay, so you fill this whole thing in. You have to do a graph for me using your preferred software. Okay, like if you have Microsoft Excel, excellent. I know you use that in your computer class, right? Okay. And if you don't have Excel, you actually can get a free copy as a Centennial student. I don't know if a lot of you know this. Like, let me just show you quickly. Okay, like if you go to I think as a student you can get a free copy directly from Microsoft as well. But if you log onto your My Centennial, there should be an eStore offer, Microsoft eStore. You just click on that. And you are students, so you can click on that, right? And there are all these softwares that are available to you, some of which are free. And I think you can just find the Microsoft one here somewhere, maybe. I don't know, maybe they took that away, maybe not. Let me just search Office. There's a lot of stuff here. Microsoft 365, is it this one? Okay, maybe they took it away, but I think it's included in your student package already. Okay. But anyways, if you can ask your computer teacher, he probably will know where to find it. But if you don't have it, that's okay. You can use Google Docs, right? Okay, like if you have a Google account, you can just click on the waffle icon here, and there's supposed to be
It's Google Sheets, I think.
yeah, maybe I just do that. Thank you. Google Sheets, right? And then you just click on it, and then go to Sheets, and then you can use your spreadsheet to do it, right? Okay, right here, Sheets. Sorry, on the side, blank one. Okay, so try making a graph, and if you don't know how to do it, that's okay. We can probably do it together or something on Tuesday. So then once you finish the lab, once you filled it all in, you will submit an electronic copy to me, okay, at the Dropbox. Is that clear? Does anybody have any question about what needs to be done for the lab? It's like the easiest
How would you
thing.
submit it?
So I will create a link for you, but you can go to assessment, assignment, and then there is the folder.
Okay.
Okay, I haven't opened it yet, but I will. Okay, later on, right? And it's gonna be due on, like I said, right, like October 5th. Okay. Now, any of you use Mac here? Mac computer? Anybody? Yeah. Okay, so listen carefully, you Mac users. Okay, if you're Mac users, I cannot open Mac file. Okay, I have a PC. The school only gave me a PC. Okay, so if you submit it as a Mac format, like .page, that's the format, right? .page. I won't be able to see your assignment. You have to convert it into PDF first. Okay, so you either hand it in through PDF as a PDF, or you hand it in as a document. Okay, .doc. Do you understand what I'm saying, Kiana? Like, do you know how to convert it into PDF? Okay, so that's pretty much it. All right. Yeah, if you can't figure it out, just Google it. Okay, I'm sure they have.
I think you can actually take the doc file and put it into Google Docs. Like you can import it and it'll have everything on Google Docs. So and then you can download it as a PDF. I think. Because that's what I do. I don't use WordPress or whatever.
Right. But the thing is, like, if you download it as PDF, like you cannot type in a PDF, right? For some
yeah,
people, yeah, that's.
I believe, yeah, this one though, it's a download as a doc, the Beetroot Lab.
Right. Yeah. So, but then you submit it as a doc as well, okay? Or as PDF. Okay. That's pretty much all I want to say. I mean, I have some bad examples to show you. Let me see if I have it. Lab. Beetroot lab. Oh, there it is. Okay. Like I'm showing you these, not to like, shame people or whatever. Like I'm just showing you so that you don't do it, okay? Like don't do this, okay? Type your answer, okay? Don't write it. I mean, I can still read it, but then really, right? Like this is college. You should, submit work that are a little bit more, presentable. I also have people who hand it in, like this. Like don't do it, okay? Don't hand it in on a separate piece of paper. Just do it directly in the worksheet, right? In the Word file, so I can mark it directly. Okay. What else do we have here? Okay, the graphs. Okay, this is not a good graph because there's no axis and there shouldn't be two lines. Your graph should only have one line. Again, if you watch the video, the lady in the video actually tells you what kind of graph to create. Okay, so that's it for now. In terms of the lab, again, any quick questions before I move back to the lecture?
Can I work on this lab like right now? Or,
You
like,
can work on it anytime you like, but
Okay.
just don't hand it in yet. Because Tuesday I'll give away more answers, right? Yeah.
But it's not open, right?
The lab is open. You can download the lab, but you just can't submit it yet.
Unfortunately.
Okay.
And this
Okay.
assignment,
Mm-hmm.
Endoplast, this assignment is already available?
Yes, it's already available under if you go to content and if you go to virtual labs. Okay.
When will you open
The submission,
Yeah.
maybe after class. Yeah.
Okay.
Yeah. But again, don't submit yet. Okay, guys, don't submit yet.
No.
Even if you think your answers are all correct, just wait until after Tuesday's class, see if there's something that you miss. All right. All right. It's 10%, right? The lab. We have 2 labs together. They're 20%. They are an excellent way to boost your grades if you didn't do so well on the first quiz. All right, but you got to put in the work to do it. All right, I think I talked too much already. Let's go back to our lecture notes. So why do we care about these hypotonic, hypertonic situations, right? Well, I imagine a lot of you trying to go into the healthcare sector, right? Some of you are trying to go to the practical nursing program, paramedics and whatnot. And in those professions, you sometimes have to give IV to your patients, right? Okay, IVs are intravenous solution, right? Fluid resuscitation, exactly. So typically speaking, we give people normal saline. Okay? That's the most common kind of fluid to provide for someone who just needs a little bit of extra fluid. This is isotonic, okay? 0.9% saline. 0.9% means there are 0.9 grams of sodium chloride for every 100 mils of the solution. Okay, so that's the level that's isotonic to the cell. And we give it for people who need hydrations, right? So people who suffer from excessive bleeding, hemorrhages, right? Vomiting, diarrhea, and they just need a bit of fluid, right? So that is that. I always like to tell this story. When 1 week after I got married, my wife was cooking the first meal, right, for me, which was, a nice thing, right? But then I got severely food poison from it. She was fine. She didn't get food poison from her own food. I did. And I had to go to the hospital, the ER, because I was just like vomiting so much. I couldn't keep anything down, not even fluid, right? And so they have to give an IV for me. And I remember at the time that the nurse was just doing the calculations on her glove. right? She had a Sharpie and then she was just doing the math there. And I remember sitting there looking at her. I was like, man, why don't you just use a calculator? Like, are you doing it right? Like, what if you miss a decimal point or something like that, right? Like, it's gonna, give me the wrong fluid, right? But turns out, it's fine. She know what she was doing. And I guess that's what you guys are supposed to do, right? Like in your math class, your teacher probably tell you to practice your mental math, right? Not to use your calculator too much, because you never know, right? You never know. Sometimes you might have to do some bedside calculation right on the spot, and that could come in handy. Okay, but I needed rehydration because I couldn't keep the fluid down. Now, in some cases, we want to give lower concentration saline. Okay, that's 0.45%, and that is a hypotonic solution. Right, it's more diluted than your cells, and that's useful for treating things like hyper. That's too much. Natri, which means sodium. That's why sodium is Na, right? Emia. Emia is in the blood. So this is too much sodium in the blood. And that could happen because of severe dehydration, not just your typical, oh, I forgot to drink water for half of the day. This is like, if you save someone from who's trapped under the rubbles or something for a couple of days, where they're like severely dehydrated, then you need to fix the hypernatremia. People who are diabetics, they sometimes get what's called ketoacidosis. In diabetes, your body thinks you are starving, even though you're eating, and it switched to metabolizing fat. And one of the byproducts of fat metabolism is ketone. And these ketone builds up in the body. It causes the blood to become acidic. And you kind of want to wash them off. You want to dilute them. And that's when you would give them a hypotonic saline. Okay, this is half the concentration of normal saline, right? Sometimes you want to give them 3%, okay, 3% saline. And this is used to treat hyponatremia. This is when you don't have enough salt and you want to concentrate your blood a little bit. That's why you would provide extra sodium for the situation. Okay. Actually, why does this show up as oh no, sorry, I was reading it wrong. This is hypertonic. That's right. Okay. So those are the different kinds of fluid situations that you need to for different scenarios. All right. Now we're going to talk about a different kind of transport. Right, finally, we know about diffusion, we know about osmosis, but there are other ways for things to get in and out of the cell. And depending on whether or not it requires energy, we would classify it as active transport or passive transport. Passive transport does not require energy, okay? And it's just the movement of molecules down their concentration gradient. Okay, they naturally go from high concentration to low concentration. You don't need to do anything to encourage the process. It just happens on its own. Just like a ball will roll down the hill, no problem. Active transport, on the other hand, requires ATP. Okay? You need to supply energy for it to happen. And this involves moving molecules against their concentration gradient. You are taking them from an area that's low concentration to an area that's high concentration. Okay, so it's like pushing uphill, right? It's tough. It takes energy. Okay, passive transport, high to low, and it's easy, no energy. Active transport, low to high. It takes energy. So let's start with passive transport. Okay, passive transport. We already learned about diffusion, right? That is just the blue dye that I was telling you about, right? The Febreze, right? The perfume, it naturally spreads, right? In a cellular environment, things that are small, things that are hydrophobic, they have no problem crossing the cell membrane through simple diffusion. Okay? If they are small, like water, they could just pass through the gaps in the phospholipid bilayer. In some cases, if it's hydrophobic, they would just dissolve in the hydrophobic tails, and then it will pass right through, no problem. Okay, so things like H2O, carbon dioxide, oxygen, and some steroid hormones, they will go through. Osmosis is the diffusion of water, right? Okay, so water can pass through on their own. So all these things will just happen without energy input. Sometimes the molecule is too big, right? So like glucose is relatively big compared to some of the things we talk about, the oxygen, the water. And sometimes the particle is charged. It's either positively charged or negatively charged. And the phospholipid bilayer does not allow these molecules to pass through easily. Okay, if it's too big, it can't squeeze through the phospholipid, right? If it's charged, then the hydrophobic layer will block it. And so in these cases, the cell are going to need to use what's called facilitated diffusion. Okay, so facilitated diffusion is still a type of passive transport, but it will require the help of a transport protein, a membrane protein. The molecule is still going from high concentration to low concentration, but it needs a little bit of help to pass through the membrane. So the help comes in two forms. Okay, we have what's called channel protein, and as you can tell, this is an integral protein, right? This is integral protein, and it creates a tunnel, a corridor for the material to pass through. Okay, these are typically ions, they are charged particles. So by creating a tunnel, it shields the charged particle from the hydrophobic environment. Okay, so it just passed right through without having to interact with the hydrophobic tails. Okay, now in this case, they are coming in, but if the concentration switches, if it's more concentrated on the inside and outside, then the direction would also switch. Okay, the protein in this case is not dictating the direction of movement. They are simply there to provide a way for things to pass through. Okay. The other kind of facilitated diffusion is what we call a carrier protein. A carrier protein, instead of providing a tunnel for things to go through, this is more like a revolving door. One of those revolving door, like at the mall or something, right? Like the molecule would get in and then it would turn, and then now the opening would be facing inside, and then the molecule gets off. Right? So these kind of carrier proteins are more suitable for transporting really bulky molecules, things like glucose. Okay, so in the channel protein, the protein is just sitting there. It doesn't change shape. It doesn't do anything. There's a hole that let things pass through. But not anything, right? Not just anything can pass through. You have to have the right, you have to fit, right? It's like that toy that kids play, babies, not kids, baby plays. Is it called a shape sorter, right? Like they have all these openings in it. You have to fit the hole to get through, right? Okay, and that's the same case with the channel protein, right? It's only designed for a particular kind of particles. Over here, the situation is a little bit different. The protein will grab onto the molecule that you're trying to move, and then it will change shape, and then now it opens on the other side and spit it out. Okay, so there's some kind of change in shape, subtle change in shape that translocate, that moves the molecule across the cell membrane. So the permeability of the cell membrane is affected by various things. We already discussed this earlier, right? The membrane composition. If you change the amount of saturated fat, if you change the amount of cholesterol, that's going to affect the fluidity. Right? If you have more cholesterol, it will become more fluid and therefore it will become more leaky, right? Things like temperature, pH, ionic concentration, and the kind of environment that it's in will also affect the permeability. And for the Beetroot lab, you will be specifically looking at how temperature is going to be affecting the permeability. Okay, so the beetroot has a very vibrant color, right? The purple color that's very distinctive to the beetroot, and the beetroot colors are contained within the cell. But when you disrupt the membrane permeability, when you make it more leaky, then the pigment is going to come out. Okay, if you ever made beetroot soup before, the whole soup is, you know, pinkish, right? So you can imagine like what the heat is doing to the beetroot. And afterwards, again, I'm talking about making the soup. Afterwards, if you fish out the beetroot, it would be colorless because all the color actually leaked out, right? So, you know, just thinking about that scenario, you can probably have a pretty good hypothesis on how temperature is going to affect the permeability. Okay, and again, you don't have to explain why in the hypothesis. You just have to tell me how it will affect it. Okay. On that note, you have to be careful. Like people sometimes write this. Okay, they write temperature, higher temperature will affect the membrane permeability. That's what they write. Okay, higher temperature will affect the membrane permeability, and then I give them 0 for that question. Why is that not a good hypothesis? What's missing in that hypothesis? Let me say it one more time. Higher temperature will affect the membrane permeability. Why am I not giving them any marks? Am I just a mean person or what's going on?
What kind of temperature? How high?
How high? Okay, perhaps. Anyone
I don't
else?
think there was a lot of details into that statement.
You don't think there's a lot, not specific enough? What not specific enough? What is missing specifically? Here, let me type it out for you guys to read it. Higher temperature will affect the membrane permeability.
Osmosis system is missing.
What system is missing?
Osmosis.
Osmosis?
Yeah.
Not quite. Doesn't mention. That's right. That's the problem. Roy, you know, nailed it on the head. Doesn't mention if it's more permeable or less permeable. Like higher temperature will affect the membrane permeability. Like what makes it more permeable, less permeable, right? You have to take a position, right? Okay. And that's why you guys also lose marks in the quiz. Some of you just write driving with the window down will affect fuel efficiency. Like how is it gonna make it more efficient or less efficient, right? Okay, so you have to take a stand, you have to take a position. Does that make sense? Okay, so I think that's where I'm gonna stop. I mean, we have still a little bit to go. It's just gonna take us probably like a half an hour, 45 minutes on Tuesday. But are you clear? Do you all understand what you need to do between now and next class? You don't have to hand in anything, okay? But you gotta watch the video. I'll post the link again in the announcement, and you gotta start doing the lab, okay? Start doing it, and then when you're stuck on some questions, Tuesday is the time to ask. All right, okay. Thank you. See you all. Have a good week. Bye bye.
Thank you. Have a good one.
Yeah.